In this lecture, we are going to cover Mendelian genetics. Before the lecture begins, make sure you've read the assigned reading in your textbook and you've made an outline of this lecture in your notes. During the lecture, minimize all distractions by turning off music, listening in a quiet space, turning off all TV or movies in the background. Turn off any extra devices as well. During the lecture, take plenty of notes, making sure to put the slides in your own words to help maximize your learning. In the next few slides, we're going to discuss Mendelian genetics. We're going to look at Mendel's experimental method, his laws, as well as human traits and Mendelian inheritance. Who is Gregor Mendel? He was an Austrian monk with a background in physics and mathematics. He is best known for his genetic experiments with pea plants. With these experiments, he discovered the basic principles of heredity, and his contributions have earned him the title as father of genetics. Of all the plants on Earth, why did he choose the pea plant? There are several reasons. First, there are many varieties, each with distinct features or characteristics, such as flower color and variations of each of these characteristics. We refer to these characteristics as traits. Another reason Mendel chose the pea plant was he could control the mating of the plants. The third reason that Mendel chose the pea plant was because he was able to track the traits with only two distinct alternate forms. He used varieties that were true breeders in that they produced offspring of the same variety whenever they were self-pollinated. Here on the slide, you see an image of the pea plant. This is self-pollinated, and this self-pollination is possible due to the presence of both the male stamen and the female, the stigma. So let's discuss the basic methods that Mendel used in his historic experiments. Mendel would make two contrasting true breeding varieties in a process called hybridization. The true breeders are defined as the P generation, or the parent generation. The hybrid offspring produced from the P1 generation are called the F1 generation. When the F1 individuals self-pollinate or cross-pollinate themselves with other F1 hybrids, the offspring that are produced are considered the F2 generation. So using this basic method, Mendel looked at the inherited patterns of several traits of the pea plant. Let's look at some important vocabulary that will be necessary for our knowledge of genetics. A homozygote organism contains two identical alleles for a character. These two alleles could be dominant or they could be recessive. Homozygotes are considered true breeders. Heterozygote is an organism that has two different alleles for a gene. They may have one dominant and one recessive. These are not true breeders. Like we'll look at in Mendel's experiments, the effects of dominant and recessive alleles do not always reveal itself in its genetic composition. Therefore, we always distinguish between an organism's phenotype, or its physical appearance, and its genotype, or its genetic makeup. So how can we determine if the genotype of an individual with a dominant phenotype is either homozygous or heterozygous? To do this, to identify the genotype, one can do a test cross. This is essentially breeding the unknown individual with a homozygous recessive individual, and if any of the offspring display the recessive phenotype, then the mystery parent must be heterozygous.
A Punnett square is often used to better understand the patterns of inheritance associated with Mendelian genetics. Remember that each parent in a genetic cross is diploid, meaning that they have two copies of each trait. During meiosis, these traits separate into individual gametes, either egg or sperm cells. In a Punnett square, the outside of the square represents the possible gametes that may be produced by each parent. In our example, a plant that is heterozygous for flower color produces gametes that either have the purple or big P allele or the white or little p allele. The area within the Punnett square represents the possible zygotes or offspring produced by a combination of a specific egg or sperm cell. Note that we cannot predict which egg cell will be fertilized by which sperm cell, so a Punnett square indicates all of the possible combinations. A Punnett square not only allows us to visualize the potential offspring from a cross, but also allows us to calculate the probability of obtaining an offspring with a specific phenotype or genotype. Notice that, assuming both parents are heterozygous, that there is a 3/4 chance that the offspring will have the dominant purple phenotype, and a 1/4 chance that the offspring will be white. Punnett squares are useful for quickly visualizing the results of a single trait or two-trait cross.
So one of the first experiments Mendel conducted was using the method of a monohybrid cross. So let's look at the specifics. A monohybrid cross is a cross where you follow the inheritance patterns of a single trait. Mendel crossed contrasting true breeders with white and one with purple pea plants. The F1 hybrids produced from this breeding were all purple, and this was quite surprising to Mendel. To further understand the inheritance pattern, Mendel then crossed or self-pollinated the F1 hybrids to form the F2 offspring. Some of these F2 offspring had purple flowers and some had white. Specifically, 705 plants had purple flowers and 224 plants had white flowers. And when you reduce these numbers of the ratio of purple to white flowers, it's about 3:1 in the F2 generation. How is it possible that all the plants produced in the F1 generation were all purple, and then they were cross-pollinated and white flowers appeared? Mendel reasoned that the F1 generation had the heritable factor for the white flowers, but this was hidden or masked by the presence of the purple flower trait. He therefore called the purple flower color a dominant trait, and the white flower color was a recessive trait. When the white flower trait was not diluted, it reappeared in the F2 generation offspring. Mendel observed the same pattern of inheritance and 6 other pea plant characters represented by 2 traits. You can find them all listed here in the table. Keep in mind that during this time, the idea of genes was unknown. Therefore, this factor that Mendel was tracking was actually genes. He ultimately developed a model to explain the 3:1 inheritance, which was produced by the F2 generation. Mendel's model has 4 related concepts that make up the model. But before we get to this, I want to point out the different characteristics that Mendel studied. He looked at flower color, seed shape, pod color, flower position, and stem length. And he looked at each of these dominant and recessive traits for each of these characters in generations and generations of pea plants. Now we're going to discuss dihybrid crosses and how Mendel used these for his various experiments. Mendel identified his second law of inheritance by following 2 characteristics at the same time. By crossing true breeders for 2 different characteristics, you'll produce dihybrids in the F1 generation that will be heterozygote for both characteristics. So by looking at the example in the pea generation, a plant dominant for yellow round seeds was crossed to a plant recessive for green wrinkled seeds. The F1 generation resulted in plants that had seeds that were heterozygote yellow and heterozygote round. When they were cross-pollinated, the offspring produced a predicted phenotypic ratio of 9 to 3 to 3 to 1 due to independent assortment. This type of cross can be used to determine whether two characteristics are transmitted to offspring as a package or independently of one another. The result of his dihybrid cross is the basis for the law of independent assortment, which states that each pair of alleles segregates independent of one another during gamete formation. This law applies to genes on chromosomes that are not homologous, or those that are far apart on the same chromosome. This is important because genes that are located near each other on the same chromosome tend to be inherited together. So before we move forward, let's look at the dihybrid cross that Mendel used.
A dihybrid cross illustrates the potential results from crossing individuals that differ in regard to two traits. In this case, the parents, or P generation, differ in both pea color and shape. A plant homozygous for wrinkled green peas is crossed with a plant homozygous for round yellow peas. Because both parents are homozygous for both traits, only gametes coding for wrinkled green peas or gametes coding for round yellow peas are produced. A diagram of the cross between two members of the F1 generation, called a Punnett square, is shown here. When the gametes from these pea plants are combined, all of the offspring in the F1 generation are round and yellow. This is because the traits for round and yellow are dominant over the traits for green and wrinkled. Because all offspring within the F1 generation are heterozygous for both traits, both the males and females can produce 4 different gamete combinations: round yellow peas, wrinkled yellow peas, round green peas, and wrinkled green peas. A Punnett square is created to determine the F2 generation that results from this cross. Of the offspring produced within the F2 generation, 9 are round yellow peas, 3 are round green peas, 3 are wrinkled yellow peas, and 1 is a wrinkled green pea. In other words, a 9 to 3 to 3 to 1 ratio exists within the F2 generation. Mendel was able to use this observation to determine that factors assort independently of other pairs of factors. This has become known as the law of independent assortment.
So these concepts from Mendel's experimental method, both his monohybrid and dihybrid crosses, gave the experimental evidence necessary to detail Mendel's laws of inheritance. Mendel's first law states that alternative versions of genes account for variation and inheritance characters, for example, like the gene for flower color, exist in 2 versions, one for the purple flower and one for the white flower. When there are alternative versions of a gene, these are known as alleles. Mendel's next law states that the 2 alleles for heritable characters separate or segregate during gamete formation and then end up in different gametes. Therefore, an egg or sperm will ultimately get one of 2 alleles that are present in an organism. This segregation of alleles corresponds to the distribution of homologous chromosomes to different gametes in meiosis. The law of segregation accounts for the 3-to-1 ratio of the phenotype that Mendel observed in the F2 generation of his monohybrid crosses. You can predict the results of a genetic cross between individuals of known genetic makeup by using a Punnett square. In the Punnett square, capital letters are used to represent a dominant allele, and lowercase letters represent a recessive allele. In the example shown, the big P is for the purple flower, whereas the little p is for the white flower. Mendel's next law states that if two alleles at a locus differ, then one of the dominant alleles determines the organism's appearance or phenotype, and the other, the recessive allele, has no noticeable effect on the appearance or the phenotype. Using the flower example, the F1 plants have purple flowers because the allele for the trait is dominant to the white flowers. Mendel's next law is known as the law of independent assortment. This law states that for each character, an organism inherits two alleles, one from each parent, and these alleles segregate independent from each other. Keep in mind, these alleles can be the same, such as the case of the P generation true breeders, or they could be different, like the F1 hybrids. Mendel made this conclusion without knowledge of chromosomes or their existence. Now that we have a basic understanding of the experiments that Mendel conducted and the laws that resulted from the evidence from those experiments, let's look at some of the traits that follow a Mendelian pattern of inheritance. Most of our understanding about human diseases and disorders come from studies conducted on model organisms, such as rodents, zebrafish, flies, and worms. This is because humans are not good subjects for genetic research. The amount of time it takes for us to produce offspring is too long. We don't bear enough organisms during pregnancy, and of course there are a lot of ethics surrounding appropriate experiments that you can do in human subjects. So even these basic reasons, which explain why humans are really bad subjects for genetic research, basic Mendelian genetic endures as the foundation for human genetics. So even though humans are usually a really bad model system for studying Mendelian genetics, scientists can use something known as a pedigree to look at genetics in human families. Human pedigrees are family trees that describe the interrelationships of parents and children across generations. This can look at inheritance patterns of particular traits and diseases, and they can be traced and described within the pedigree. So pedigrees can also be used to make predictions about future offspring. Here is an example of a basic pedigree. The white square represents a male, and the white circle represents a female. A purple square represents a male with a trait, and a purple circle represents a female with a trait. A horizontal line between two shapes indicate a mating pair, and a vertical line indicate offspring produced from mating pairs. These offspring will be listed in birth order, with the firstborn being on the left. If you look at the pedigree for a widow's peak, in the first generation, you see the grandparents. You'll also notice there are two grandparents that carry the trait, a grandfather from one set and a grandmother from the other. The offspring produced from these pairs resulted in several heterozygote individuals with the trait. So as we can see from this pedigree on the left, widow's peak is a dominant trait. Now if we look at the pedigree on the right, we can look at the inability to taste PTC. At the grandparent generation, only the homozygous recessive individual has the trait. In the offspring produced, you see the same trend of the homozygous recessive individuals being affected. Therefore, the trait can be considered recessive. There are some genetic disorders that are inherited in a recessive manner, and they can range from mild to being very life-threatening. These disorders only arise if individuals homozygous for an allele, therefore carriers of the disorder, are usually heterozygous for the recessive allele, but are phenotypically normal. Keep in mind that most people who have recessive disorders are born to parents who are a carrier of the disorder. Recessive alleles that cause disease are rare because of the low chance of two carriers reproducing. Consanguineous mating is mating between close relatives, and this increases the chance of offspring having rare alleles. Most societies and cultures have laws and/or taboos against these types of marriages. In the following slides, we will discuss some of the disorders that are inherited in a recessive manner. Cystic fibrosis is the most common lethal genetic disease in the U.S. It affects 1 out of every 2,500 people of European descent. Mutations in the cystic fibrosis allele are the result of a mutation in the chloride transport channel gene CFTR, which is found on chromosome 7. This leads to a defect or a loss of the channel, causing chloride ions to build up outside the cell. Symptoms of this disorder include a thick mucus buildup on internal organs and abnormal absorption of nutrients in the small intestine. Another recessive disorder is sickle cell disease, or sickle cell anemia. This disease affects 1 out of every 400 African Americans or people of African descent. The disease is caused by the substitution of a single amino acid in the hemoglobin protein in red blood cells. In homozygous individuals, all hemoglobin is abnormal, and by abnormal I mean that they have a sickle shape. The red blood cells in affected individuals have more of a crescent shape instead of a round or circular shape. Symptoms can include physical weakness, pain, organ damage, and even stroke or paralysis. Heterozygotes simply carry the trait, and these individuals are usually healthy, but in some cases they do suffer some of the symptoms. About 1 out of every 10 African Americans have the trait, which is actually a really high frequency for our population. Heterozygotes are less susceptible to malaria, which is actually a benefit to being a carrier of the trait. Although 2 sickle cell alleles are necessary for an individual to manifest full-blown sickle cell disease, the presence of 1 sickle cell allele can affect the phenotype. Thus, at the organismal level, the normal allele is incomplete dominant to the sickle cell allele. At the molecular level, the two alleles are co-dominant, but normal and abnormal sickle cell hemoglobin are made in heterozygote carriers, who are said to have the sickle cell trait. Heterozygotes are usually healthy, but some suffer some of the symptoms and can have long periods of reduced blood oxygen. Now let's look at dominantly inherited disorders. These disorders are rare and tend to be lethal. They arise by mutations of dominantly inherited traits. Here's an example. The example on the screen is arachnoidroplasia. This is a type of dwarfism caused by a rare dominant allele. If you look at the Punnett square on the screen, you can see how one can inherit this disorder. The Punnett square on the screen shows that the father, up on the top portion of the Punnett square, has the dwarf phenotype, big D, little d, and the female does not, both little d's. When you cross someone who has the dwarf phenotype and they are heterozygote, and you cross them with someone who is not, then you have a 50% chance that the child of this mating will have this dwarfism disorder. Another dominantly inherited disorder is Huntington's. The timing and onset of the disease significantly affects its inheritance. Huntington's disease is a degenerative disorder of the nervous system. The disease has no obvious phenotype until about 35 to 45 years of age, meaning that individuals can be completely normal physically until they reach this age range and begin to present symptoms of the disorder. Once the deterioration begins, the condition is irreversible and often leads to mortality. There are other factors that influence one's ability to inherit a disorder. Many diseases such as heart disease, diabetes, alcoholism, mental health, mental illness, and cancer are all factors that influences one's ability to inherit a disorder. There are also genetic and environmental components. Therefore, lifestyle can play a tremendous effect on whether one presents with the phenotype of a disorder. This is truly the case in a lot of cardiovascular conditions. So these conditions that are impacted both on genetics and environmental components are known as multifactorial disorders because both the genetics and the environmental factors play a role in the phenotype. Oftentimes, prospective parents are going to plan out a family or considering having a family can meet with a genetic counselor and talk about the risks for their children for such specific diseases. Each child represents an independent event, and in that sense, the genetics is unaffected by the genotypes of the older siblings. So in this lecture, we looked at Mendelian genetics. We looked at Mendel's experimental method, Mendel's laws, and the human traits and Mendelian inheritance. If you have any questions remaining from this lecture, feel free to email your professor, to come to office hours, or to bring your questions to class. Additionally, you can reach out to your learning assistant. You want to take the notes that you've made during this lecture and combine them with the notes that you made when you read the assigned portion of your textbook. To prepare for the quiz and for the test over this material, use the assisted reading questions as well as the questions at the end of the chapter and the questions at the end of each chapter section to quiz yourself.